California, 1982. Since the discovery of nucleic acid from salmon sperm by Friedrich Miescher in 1872, nucleic acid has been found in all living cells. It is now known that both types of nucleic acid occur in all cells, whether plant, animal, or bacteria. The composition of DNA is characteristic to any given species, and the amount of DNA inside each cell is strikingly constant for all different types of tissue within a species. The experiments done by Hershey and Chase have proved that DNA is the hereditary agent. It's also possible that it is connected to the mechanism for protein synthesis in cells.
Rosalind Franklin, King's College, London, 1952. After receiving my doctoral degree in chemistry from the University of Cambridge in 1945, I traveled to Paris to study X-ray crystallography techniques. These techniques allow one to view the three-dimensional structure of molecules. I've been here at King's College for the past 3 years, attempting to photograph DNA using these X-ray crystallography techniques. After I got back from my summer holidays, I had a terrific crisis with Wilkins at the lab, which nearly resulted in my going straight back to Paris. Since then, we've agreed to differ, and work goes on. In fact, quite well. See, this last picture I took, it turned out wonderfully. I must get to analyzing it at once to see if I can deduce anything about the structure of DNA. Let's see. Absorption bonds corresponding to symmetrical and anti-symmetrical vibrations indicate the equivalence of the bonds. However, failure to exchange bases at the minimum temperature indicate that the
May I ask what you're doing?
No, it would be a bother.
Well, what are you doing?
Hello, Wilkins. I am attempting to calculate the structure of DNA. Or I was until you came in.
Well, I'd like to remind you I'm also a member of this lab. I came here to get some work done, but if you're going to be this bothersome, I suppose I won't accomplish much.
Fine, I'll leave. I'm just about done here anyways, and I can't stand your presence.
What's this? DNA. It must be. You know, for someone who's so incompetent in general, this is an astonishingly good photograph. I must take it to Crick and Watson at once. They've been deep in their work, working to discover the structure of DNA, and they'd be delighted to have such a detailed photograph as this one. It's a shame Rosie went off her head and left the lab already. Now it's in use by no one. Well, I have important things to be getting to.
Oh, this is thrilling! Crick, we've done it! We've discovered the secret to life!
Yes, Watson, we have. We have to publish our findings immediately. Everyone will be thrilled. Look at this.
Wonderful indeed. I don't believe it would be an exaggeration to say that this is one of the most important discoveries of the century.
It's difficult to say since the century is a little more than halfway through, but I'm inclined to agree with you. Although you must admit, we could not have come this far without Franklin's photograph.
Indeed, her photograph was quite brilliant, but I'm not sure that she deserves credit when we publish.
Indeed. Although Wilkins seemed against her overall, from his accounts it was as if she didn't understand anything about the actual structure of DNA. She had all that 3D data for 9 months and she still couldn't fit a helix to it. All she did was produce a quality photograph. She never put it to good use.
Yes. She simply had a clue she didn't know how to interpret. I don't think her name deserves to be on the paper.
Agreed. Her image wasn't confidential. I don't think she's worth mentioning in the report at all.
Stockholm Concert Hall, Sweden, December 10th, 1962. Dr. Francis Crick, Dr. James Watson, and Dr. Maurice Wilkins, your discovery of the molecular structure of the deoxyribonucleic acid, the substance carrying the heredity, is of utmost importance to our understanding of one of the most vital biological processes. Practically all the scientific disciplines in the life sciences have felt the great impact of your discovery. The formulation of the double helical structure of the deoxyribonucleic acid with the specific pairing of organic bases opens the most spectacular possibilities for the unraveling of the details and the control and transfer of genetic information. It is my humble duty to convey to you the warm congratulations of the Royal Caroline Institute and ask you to receive this year's Nobel Prize for Physiology or Medicine.
New York Times, 1963. The prospect is that in the next few years, humanity will understand and be able to control, at least in part, the fabulously intricate mechanism through which each species of living organism transmits its essential properties to the next generation. The danger exists that scientists will make some of these godlike powers available to us in the next few years, well before society, on present evidence, is likely to be even remotely prepared for the ethical and other dilemmas with which we shall be faced.
Gene Sequencing Laboratory, United States, 2019. Hey, did you hear about that scientist in China who genetically modified babies?
Yeah, I read a few articles about it, plus I watched a video posted by the scientist who did it.
Yeah, that was super cool, although I was kind of shocked that something like that had happened. Like, I'm not even sure if it was ethically right or wrong.
Right? I totally agree. You know, this one lecture I went to was talking about how we could cure all sorts of genetically based diseases through genetic modification. But then you have to consider whether people should be able to modify their offspring before they're born because they don't have a say, you know? And then you get into the rest of
Yeah, that's enough philosophy for me today.
Right, sorry. I just get really fascinated by this sort of thing, you know?
Yeah, that's fair.
But we wouldn't have come so far without Watson, Crick, and who was that other guy who shared the prize with them?
Wilkins.
Yeah.
Although I still feel like it's a complete tragedy that Franklin didn't get any of the credit.
Who was that again?
Oh, Rosalind Franklin! You don't remember her? I'm sure her name would have showed up in some biology textbook. She was the one who took that photograph that Watson and Crick used, and it was critical to their discovery. Without her, they wouldn't have had their triumph.
Huh, and she wasn't credited at all? Really?
Yeah. Well, see, Wilkins, her lab partner, had taken the picture from her because they didn't have a very good relationship with each other. They were on quite poor terms. And as a result, Watson and Crick didn't think too much of her either.
It's really too bad.
Yeah, a real tragedy. But you know, in 2002, the British government actually created an award named after her, and it's all about recognizing women in science. Oh, goodness, look at the time. We'd best get back to work.
Right. You know, it's kind of funny how we're here talking about the discovery of the structure of DNA, and here we are only a lifetime later sequencing people's genomes for a living.
Yeah, none of this would have been possible without the discovery of the double helix. Technology's come so far. That is the end of our performance.